I'm the Associate Director of Graduate Admissions here at the Halmos College of Arts and Sciences. I want to welcome everybody to today's virtual lecture series, through the Halmos College of Arts and Sciences. But we'd like to introduce our presenter, Dr. Joe Lopez. Joe's a longtime NSU professor in the Department of Biological Sciences here at Nova Southeastern University. He has written and contributed to various publications, including journal articles, edited collections, and books. He has also presented on a wide range of subject areas, such as marine biodiversity, microbiome, stormwater treatment areas, genomics, and dynamic ecosystems. Today's presentation, in large part, is going to be an informational webinar regarding applying and advancing modern biotechnology and genomic sciences for a better understanding of marine organisms and their functions. At this point, I would like to turn it over to Joe. I'm available for questions with Assistant Director Nicole Chavez after the presentation, especially if you're interested in our 12-credit computational molecular biology certificate as well. But Joe, turn it over to you. Thank you again.
Great. Yeah, thanks, Matt. Thanks for that introduction. Thanks, Nicole, and for your organizers for this talk. Pleasure to be here. I am going to go ahead and share my screen. It's a great opportunity to talk about our research. And that's it. Yeah. Yes, it is a great opportunity to talk about our research in my lab. As Matt says, I've been here 13 years now, which goes very fast. 2007, I joined Nova. And what I'm going to talk about today is just kind of give you kind of a glimpse of the research with a lot of student participation. I mean, I get this opportunity to talk about the work that's been done by all these great students. And I think you see this is an opportunity. And I feel very lucky to have been able to apply genomics and molecular biology to a wide array of different marine questions, which I hope you'll see today and kind of get that snapshot of what we're doing. It's an exciting field. And I want to convey that to you, that there's just a lot going on. It's an interdisciplinary field. And we look at it from the genetic level. So again, my background is in evolutionary genetics and biology. And I decided to turn this training and the methods to the ocean and various habitats there like coral reefs and the deep sea. But my main background is genetic work, which is lab-based, but the tools and the technology have just really advanced in quantum leaps, really, for the 20 years or so, 25 years that I've been doing research. So it's amazing just to be able to witness and be part of this revolution. Really, it's not an overstatement. We are able to read whole genomes of organisms. We can read the whole hereditary material, which can be millions of base pairs in a bacteria to billions of base pairs in a multicellular organism or eukaryote. So that's the tool that you'll be hearing as a thread throughout the talk. And another one of my interests is understanding the complex relationships between organisms. So that's not just the macroorganism, the host that you see easily with the visible eye, but the other organisms, the smaller microscopic organisms that live on and inside almost every living thing, that there's an association called symbiosis. And this is very well exemplified in the marine invertebrates, which I started focusing on as a postdoc in Nancy Knowlton's lab in Panama. So that's going to be the thread of the work here that I'll cover throughout the talk, touch base here and there. Again, the background is in genetics. with a focus on diversity, biological diversity, which has various types of strata in terms of, you can look at the genetic diversity, organismal diversity, ecosystem diversity, but they are all linked. And genetic diversity is the basis of the speciation that we see. So I've tried to apply those techniques to understand the association of symbionts, phylogenies, which means relationships, genealogical relationships between organisms, such as marine sponges. We can also apply those to the microorganisms that live free living in various habitats like the water, seawater, and various other types of habitats, and or living in symbiosis. And then we could look at DNA and RNA. So those are the areas that are expressed from the genetic code. Now, one of the kind of the major divisions or foci in my lab throughout the years has been either prokaryotic or eukaryotic. And as biology majors, you know the differences here, but the prokaryotics are those bacteria, archaebacteria, the single-celled organisms that are also everywhere, much simpler, smaller genomes, and then the eukaryotic, the higher, the more complex multicellular organisms. And we know this from our Bio 1 basic biology, that we can look and divide nature into these two major groups. But luckily, again, my lab is able to look at both of these groups in tandem and in various ways because of symbiosis. Now, I'm not going to get to talk too much about this is an ongoing project that we've started just in the last year, but it is on the prokaryotic side. And we have obtained some funding to try to understand what's causing cyanobacterial blooms. These are the blue-green algae bacterial blooms that have been occurring over the past few years in Florida. But it's a prokaryotic study using genomics and metagenomics. And unfortunately, I won't have the time to go into that, but this is one of our current projects here, is to understand and apply genomics to understand what's causing these blooms. Now, Matt asked me to talk a little bit about, before I get into the research per se, some of the courses that I teach and some of the students that have worked in my lab. So this is just a slide showing one of the programs I helped develop at NOVA over the past few years. And right now it's a postgraduate certificate program focused on genomics and bioinformatics. And I teach a genomics class in this 4-course program certificate. With the main idea of putting this together was to try to give students the basic skill set to get into industry of the genomics and bioinformatics industry, biotechnology, just to get their foot in the door. And then if they wanted to develop this further, the skills, they could take more courses or go for a higher degree. But these courses of bioinformatics, genomics, and a programming course and a database course in computer sciences will be enough to sustain that entry-level technician job, for example. And I know this is a kind of an old slide. I haven't updated it, but genomics has been moving forward. As I mentioned, we're generating more and more data. So I don't think it's going to go away. The opposite, there's going to be a greater need for skilled bioinformaticians, computational biologists to understand the data and be able to work with the data and analyze it. Sometimes this takes a training in biology as well as computer science, and it can be difficult to find an expert who knows both. Myself, I don't do the nitty-gritty of a lot of the coding of what goes into bioinformatics, but for for you upcoming students, this is an opportunity for you to, I think, to pick this up. As you see, the market is there. You can go into industry as well as academia. Government jobs may increase in the future because this data is going to be there, and there's going to be a lot of utility for this types of work. I also teach a microbiology course, and it's not shown here, but I teach an intro to microbiology, which is just changes every year. So much exciting progress in microbiology. CRISPR research was based on microbiology. You saw that was a new Nobel Prize given out this year. Of course, COVID-19 is a virus, virology. We need to develop better vaccines. That's immunology, epidemiology. These are all growth fields. And if you're looking for work, I think this is where you want to apply your focus, because genomics, microbiology, there's going to be a lot of attention and need for people who come up with new ideas and new solutions. So again, there's a lot to that future ahead of us. I just wanted to show a couple of my students who have gone on to Ph.D. degrees, doctoral degrees. I've had over 20 master students through the years while I was at Harbor Branch as well as at Nova. And they've done all, again, great work. They do all this themselves through thesis research, which I help guide, but we develop theses together, look at questions, and I try to have our students do publishable research. This takes effort, it takes funding, but for the most part, we do our best to try to get work published. And these are two students who've gone on to doctorate degrees in the work. Dr. Chavez Vinegra made it onto a Chasing Coral movie a few years ago, which highlighted the catastrophe we saw in the Great Barrier Reef in 2016, where 1/3 of the reef collapsed, most likely due to global warming, sea temperature changes, and it's still in decline. Dr. Svandos here, I don't know if your name out, Karen Svandos was a master student who now does some very interesting cancer research at Johns Hopkins. I've had several students recently graduated, like Lindsay Freed, and she did some interesting work on the anglerfish. This is part of a bigger project called Deep End with Tracy Sutton as the PI. This was funded research after the Deepwater Horizon oil spill in 2010, and he was able to get funding to look at the pelagic habitats of the Gulf of Mexico, relatively understudied, but we looked at trophic associations starting from the very top, apex predators, down to jellyfish, crustaceans, and then our lab was focused on the microbial portion of that web of life and the food chain. And we found that there was a lot of structure in the Gulf of Mexico, which was very interesting from a vertical depth gradient. Well, one of the offshoots of that project was Tracy was also able to collect very inaccessible anglerfish. And he discovered a new species of anglerfish actually in this project. I'm not sure if it's the one shown here, but this is a typical anglerfish. You've probably heard of these. They live in darkness because light pretty much disappears under 200 meters. That's the aphotic zone. And so animals, a vast habitat, one of the largest habitats in the planet, really, when you look at depth gradients, they live comfortably in these cold, dark areas. So Tracy he studied multiple fish species, and this is one of them here, shown here. Well, we came about because it turns out that this ESCA, or the lure that you see shown here, is actually, see if I get my pointer going up. But the lure shown here is called an ESCA right here. And it lights up with bioluminescence. You know, that's not electricity or anything down there. It's powered by either the animal or bacteria. And we hypothesized that it was bacterial in origin. It lights up. There's some functions that go for the host, like possibly a signal for mating, or a warning signal against predators. Most likely mating, possibly because these are fairly rare species in a big habitat. And you might have heard of anglerfish in this regard. I've seen pictures of this, how some species mate. Well, this is a picture of two anglerfish. The female is the larger one, and the male is the smaller one. And what it does is it actually bites the female, attaches to it, and this is the mating process. Over time, the tissues actually fuse, and possibly due to that rarity, they're mated for life, whether they like it or not. And the male is donating its sperm there throughout the lifetime. So that's one species, and the light organs may be used for that signaling. So we thought that we wanted to look at the bacteria that lived in the lure and also the anglerfish using a technique called 16S RNA amplicon analysis. This is a small gene found in all organisms, but we can amplify them with universal primers, and then we can compare those and then identify bacteria, different species of bacteria using that gene after we read the sequences. So Lindsay did that, and this is just part of her thesis here, but each dot there represents thousands of sequences of 16S. They've been put together on this two-dimensional NDBS plot, and what it shows is a clustering. And in this case, she was able to isolate samples from water and then some from the anglerfish, also different parts of the anglerfish, but here they're all kind of grouped together. And we see that there's a clear difference, that the anglerfish bacteria have a distinct microbiota or flora that is different from the anglerfish themselves. And so this really supports the symbiotic aspects. They are different from normal seawater. Well, she went on to further characterization, looked at those sequences in more depth. And what you see here, most of the coloring in most of the abundance of the bacteria here is in pink, and that's related to the Vibrionaceae. And that's a very well-known family of bacteria that do contain photobacteria, or bacteria that emit light or bioluminescence. So that made a lot of sense. And you can see some of the ESCA here were dominated mostly by these Vibrionaceae. So she made a very nice characterization, which we published in the end, showing this identification and characterization of bacterial groups inside this anglerfish species. We've gone on in my lab to do further characterizations in other animals and other habitats. This is just kind of a glimpse of those. We've, of course, done marine sponges, I'll talk about in a little bit. We have a shark study, which is now in preparation. And we found Rachel Carnes did that study. It was a project which used the same techniques, and we found a lot of diversity in the teeth, which kind of makes sense. We've also done more studies of different water habitats, like our local reefs here off South Florida, as well as the Port Everglades Inlet. We've studied the inlets using microbial methods to look at what's in the water there. We've also done a study in collaboration with partners in Ireland. They were studying various bat species, which have right now big time in the news, a big concern. But bats, we should know more about how they do in their physiology. And the aspect that Emma Teeling was looking at is the longevity of bats. And that's an interesting aspect, because even for their small size, some species tend to have very long lives for their size, and the physiology of that and the basis of that longevity is not known. So we collaborated, and she provided bat poop samples shown there, and we were able to do some microbial analysis of that, and that was published in the Hughes paper. We've also done some human work that looked at bacteria for forensic analysis as a way of maybe individual identifications, and that has also been recently published. Now, just to backtrack a little bit before I start talking about some specific research and specific projects, is this idea of symbiosis again. And I try to kind of highlight this in my microbiology class, but overall, just if you didn't know or weren't too familiar with symbiosis, it's basically just the association of more than two organisms together. The basic is two, but there could be more species involved. And the idea is that one partner or both partners are either getting something out of it, getting a positive feedback. Maybe one gets a structure to live in. It might give off nutrients through its metabolic products, and that's where you got a win-win. Or you can have one that's just kind of neutral, and it's just using the abode as a place to live. And then you start going downhill from there, because either there's a change in the host, and if the host's immunity degrades, then that once beneficial microbe, the good bacteria or the good microorganism, starts becoming a bit more of an unwanted guest. So this happens throughout nature, and it's a dynamic process. And we just have to keep that in mind, even as we look to our own health. This is most of the bacteria I talk about in class in microbiology are beneficial. It's a very small fraction of bacteria which are actually causing disease. Oops, I had that set on automatic. So I tend to show this quote in my class, made by a very famous evolutionary biologist, Lynn Margulis. And it's very true what she says here, and I'll talk a little bit about the moon in a little bit. But we have a living planet mostly because of microorganisms. They're carrying out fundamental processes like creating oxygen or recycling elements. We could not have this, we couldn't live on this planet without their work. So that's why they're beneficial. And they create the atmosphere. And as we know, the moon has no atmosphere. So that's the big difference. So as one more kind of aside here, I did we published an opinion piece last year thinking about this symbiotic aspect of how we're dependent we are on microorganisms. And it was partially inspired by several things which came around in 2016. One was a seminar by a former NOVA alumnus, and she was working at NASA, and she was talking about this new Mars push that we want to start going back to Mars and exploring it, possibly colonizing it, which is a long-range plan. There was a movie that came out with about the Martian, and Matt Damon was stranded there. And so I wrote this article because they didn't talk about microbes, and he needed those microbes to grow the plants that he lived in. So we published this in FEMS, and it got a pretty nice response here. The regular media picked it up. Some of them kind of misinterpreted some of our words in terms of we were not currently calling for an infection of the planet Mars, but we wanted a systematic kind of approach to looking at what microbes can do for interplanetary colonization or eventual colonization, and that it had to be done in a systematic way, and that we needed to include microbiologists in this discussion of exploration. Right now, people are going full bore, like Elon Musk and Blue Horizons and all the other kind of private companies trying to be the first to get to Mars. And we probably know we can get there, but it's a long way. And it's 6 to 8 months. That's a lot of time for people to spend getting there, coming back safely. We might want to think about these other routes. I mentioned the moon. So the moon, we were there in the 70s. We made 6 to 7 trips on the moon, spent over 70 hours on the moon. And it might be interesting to see what happened to the waste products, because they were left there. They were not returned. And microbes were, so they're there. They're probably not alive, but it would be interesting to see. So we have to think about that, because we will carry our microbes with us wherever we go. And if we want to safely decide on, either trying not to pollute or contaminate a planet, we could start doing some really interesting experiments or mesocosm experiments here. Maybe we could pick the best microbe to carry with us that would produce an atmosphere or produce a lot of oxygen that's needed. Mars has very little breathable oxygen. It's mostly a CO2 atmosphere, so we couldn't survive there anyway. Whereas there are various microbes that are anaerobic. They can take that CO2 and they can convert it to oxygen. So it's a very different habitat. We need to probably do these experiments here, and that was the main thrust of or part of the thrust of our opinion paper, was to maybe take that. And so this is a new branch of research. I'm not actually actively doing it now, but I've got that on the horizon to start thinking about extremophiles, how we can maybe utilize microbial and microbial genomics, microbial research to try to maybe develop a better colonization strategy for these new extraterrestrial initiatives. So that's again in the future. Coming back to Earth again, we're on a microbial planet. There's been a slew of research the past several years trying to characterize the what's called the Earth microbiome. As I mentioned, this is a very nice review paper study that looked at the Earth microbiome across all these different habitats. So we have basically two major groups, the free living and the symbiotic. These were taken from all these different sites, from marine sites to terrestrial sites to Arctic sites to tropical sites. And they put this all together in an analysis to look at the different types of microbes that live in all of these different habitats. So this is an ongoing various types of study. I mentioned this before. But you can now, because of the methodologies and the genetics, we can now look at the whole genomes of most of these microorganisms and try to piece together what they're doing in these various habitats. Now, I want to go now and switch to the eukaryotic side. And it's part of the symbiosis, but we also are on the verge, or have been on the verge for the past few years, of now reading these much bigger genomes. So again, it's about 3 orders of magnitude larger than most average bacteria. When you want to read a human genome, for example, that's about 3 billion base pairs at 3 orders of magnitude larger than a bacterium. And the first human genome took about 20 years to finish. It's actually just recently been totally finished, but from 1989 to 2001, about 12 years to finish, but it took major effort, $3 billion to finish. And so doing a eukaryotic genome is not trivial. It took a lot of time. It created a lot of technology, but those days were difficult to do. So now people are looking at how to sequence other genomes and other eukaryotic genomes, especially non-model organisms. And this group called the 10,000 Genome 10K group looked at what species could we start looking at and sequencing the genomes. This was a brainchild of Steve O'Brien, who happened to be my Ph.D. advisor. And He was focused on cat genetics, also HIV research while at NIH, and he started the Genome 10K project. Well, he also helped kind of develop another spinoff. And when he first came to NOVA in 2012, 2013, we talked about turning the genome research to invertebrates, because the invertebrates are those animals that do not have a backbone, and yet they represent the majority of animals around the planet. And they tend to be small, hard to find sometimes in the dirt, in the water. And yet, if they're going to be part of this Genome 10K or these bigger initiatives to understand biodiversity, they're going to have to be included. So I like showing this tree from Dunn and Ryan from 2015. And we're into these trees to show relationships, but it's the major phyla of animals. There's about 30 here. And I just want to turn your attention to, you've probably seen these, just turn your attention to those histograms that are missing red. The red indicates that a organism or a phyla has a representative genome. Now, the vast majority of phyla at the writing of in 5 years ago, that's about 20 phyla had no genome representatives in the database. So that's a lot of diversity that has had no data. And it's still hard to get data for some of these phyla, but this represents a big chunk of eukaryotic biodiversity for metazoa, for the animals. So there's big gaps here. This kind of reiterates the same thing, that most of these organisms here would have been sequenced, but there's a lot that have been missing. And for that reason, we convened a maiden meeting in 2013 at NOVA. I gathered several of my colleagues and their colleagues, and we thought we'd put together a companion group to the G10K group, and it's called GIGA, focused mostly on marine aquatic invertebrates. Now, again, this is a very large group of animals. There's no way, not even our small group could cover them all, because this covers those 20, those 30 or so phyla on that tree. But you can see just by this picture the diversity in morphology that are found, from crustaceans to worms to mollusks. They tend to be, again, aquatic, but very inaccessible and underwater. Various different forms. Some phyla have just a few specialists, people who know how to identify them. So that's another trick too. We need expert taxonomists who know about the biology of the species to help us identify it. And so this is a big community effort, which is part of the reason for forming GIGA. Again, what you can see here is that GIGA can span all these different phyla. The G10K is only one phylum here, the Craniata, which includes the vertebrates and ourselves. The I5K here is the insect group, Insect 5K, and they're only one phyla, whereas all these other phyla are really understudied and underrepresented. This is a new article that just was published. Very interesting read if you get a chance. Kind of characterizes the invertebrates we're focused on as uncharismatic and icky. Nobody wants to touch them. Very few people study them, but you look at these various characteristics here, you can see the number of species just within echinoderms, the starfish, the sea stars, and the sea urchins. There's 7,300 species here and barely looked at. Rotifers, 3,000 rotifer species. Flatworms, 26,000 flatworms. And this is just kind of scratching the surface. So there's a lot of biodiversity there. It's going to require a village or a community of scientists, which is the big idea of GIGA. We've had 3 meetings and workshops over the years. We want to continue to have meetings to exchange new ideas and new methods. we want to be this kind of central coordinating group to help students in training in bioinformatics, and also limit the duplication of effort, because that is a big thing. That if you're coordinated, you won't be doing the same thing or reinventing the wheel, and that's the idea. It's not also limited to just geneticists, because we need those morphologists and those biologists and ecologists to tell us about the species. And DNA sequencing is getting cheaper. So that part is actually getting easier over the years, and we're going to think the technology is going to get cheaper. But the bioinformatics is also going to continue to grow and develop, and you still need to collect. So that's where we need these ecologists and the biologists to tell us what we're really working with in the identification. So these are all important pillars of the genome initiative. And then once we get the sequencing done, you get something like this. This is the data in the 4 nucleotides. The technology is going to just continue to be faster, cheaper. And then the big question is, how are we going to handle this data? How are we going to analyze it and put meaning to the data? So again, you can see among the invertebrates, we have some very interesting species and diversity. The hydra, for example, is an organism that is thought to live forever. It's been in culture. Some of these cultures have gone on and have not senesced. You have crustaceans and shrimps and various crabs that feed millions of people around the world. So it's a very big fisheries. So we should understand what's kind of sustaining those species, their evolution and ecology, and then have their genomes read for how they survive. The octopus, as I'll show later, one of the smartest invertebrates. It has a very well-known ability to problem solve. And then here's the kind of teddy bear of the invertebrate world. Some of you might identify the tardigrade, right? And it's been known to survive desiccation. And I think there was a crash landing of an Israeli satellite on the moon. And so I think there's tardigrades on the moon now. So I'm not even sure if they survived, but this is a microscopic invertebrate. It's aquatic, but it's an amazing organism. So again, the invertebrates encompass all this diversity. We're focused on the marine diversity, but you count all the invertebrates together and you're at over 1 million species. So just again, finding the experts and expertise to identify and tell us about these invertebrates can be difficult. The lion's share of this diversity, of course, are the insects. So that's shown here. We probably have at least 1 million insects, or 1 million insects that have been counted and named, but there's probably more of those. So we can leave those out and we still have several 100,000 marine invertebrates. So just in the past few weeks, we have been lucky to get some funding from the Gordon and Betty Moore Foundation. And this is just starting up, but they have generously given us and a group called our Hub, collaborative scientists around the world, some funding to sequence at a very high level, high quality, chromosome level genomes of 50 symbiotic pairs. And so we're going to focus on photosymbiotic pairs. There's another group that works on corals, another group that works on sponges, and another group that works on protozoans. But we're focusing mostly on photosymbionts. And this is just shown here. Photosymbionts are those that are having a pairing where you have the microbe actually being able to photosynthesize like a plant, and then the host will hold it. We see this in corals, sponges. And corals is probably the most easiest to see because they have an algae zooxanthellae symbiont that lives with the coral. What does the coral get out of it? Well, it gets what they call junk food. It gets a lot of sugar and carbohydrates. This is all converted from CO2, sunlight, and we get this reaction to get glucose and other carbohydrates. So it helps the host, the animal, gets this carbohydrate for energy. And it is found, this phenomenon is found across multiple invertebrate groups, from corals to aceals to mollusks. This is a solar sea slug, giant clams, sponges, tunicates, and we can do this comparison once we have the genome. So this is very exciting. We're very excited to start this project. We just did a collection here in the Keys with one of my new students, John Pettengill, and here you see this group called the Elysia sea slug. It's a sacoglossan. It eats algae, and then it retains the algae in its tissues and can use it for photosynthesis later on for energy. We also just finished a genome of a bryozoan, which may be the first bryozoan group for the whole genome. It's a draft genome started by Jason Kwan and written up by Michael Rako. Again, very excited to have a bryozoan genome right now on the evolutionary tree. This was very interesting because you might have heard that this bryozoan creates an anti-cancer compound called bryostatins. This bryostatin's been studied for several years, and it's a protein kinase C inhibitor, so it has potent anti-cancer activity, regulation of cellular activities, but the compound itself is produced by a bacterium, by a symbiont. So this is the kind of chain, the chain of events or the metabolic machinery that produces bryostatin, but the bryozoan genome itself hadn't been determined. So now we might be able to see, putting this information together, how they cooperate and how the symbiont is maintained over time. So very exciting, and glad we had this genome recently published. We have also other questions. Hopefully I'm not running out of time here, but Matt, I got just a couple more slides. One of the questions about carrying on sponges is, what was the first animal? And there's a big debate now whether ctenophore or a comb jelly is earlier offshoot than sponges. And that was all due to genomic data, which came out in the early 2000s. We saw that the ctenophore looked like it came down before. Was it more of a basal? So this is an ongoing debate as well, but we can get to the bottom of this with more genetic data. And again, these are sister groups to the rest of the animal groups. Very interesting. So I mentioned that sponges are kind of our main focus in vertebrate in my lab. We're still doing sponge research, which is the simplest animal. It's a filter feeder, but we have many species of sponges right here off our reef in Fort Lauderdale, many species, and we've just finished, one of my students, Yvonne Desplat, finished a gene transcript study, which came about after the Deepwater Horizon oil spill as well. We wanted to see the effects of oil on the transcription of the sponge. So we did some dosing experiments. I won't go into detail here, but we were able to carry out closed experiments in aquaria and look at the effects on gene expression on a species of sponge called Cinecarella that we're trying to develop as a model organism. And what Yvonne was able to show was that there were clear differences between a control and dose sponges for 1 hour or 24 hours. Blue dots here represent a gene or a transcript of RNA after this time period, and it's significantly expressed over the control or over basal expression. So these are twofold changes. He's identified the genes, and this is several of them. One of them is called ubiquitin. We've also found tumor suppressor genes and oncogenes, which appear to be differentially regulated or expressed either up or down, depending on what that gene function is. And lastly, is GIGA is part of a even broader group of genome initiatives, and the biggest one, which is kind of came online in 2015, is the Earth Biogenome Project. This is a bigger network of networks, and GIGA is proud to help out and assist in this, where they want to sequence 1.5 million species. This is all of the diversity, eukaryotic diversity of the planet, including those insects, including those vertebrates. It's considered a moonshot because it's so ambitious, but the moonshot helped create technology. It was, it helped further those horizons. It helped really gain traction. It helped computer technology. And so we'll see probably a lot of benefits from this initiative. And it can be likened to an almost futile event, like Mark Blackster, the leader of the Darwin Tree of Life project and part of EVP, commented, it's Sisyphean in character, which is a Greek myth where this Sisyphus was condemned to a lifetime of rolling up this large boulder up a tree, an exercise in futility because every day after he rolled it up, it would roll back down. It was a very nice analogy he made, because this is where we might feel like we are with such a monumental task of sequencing all these species. But at least, according to this article by Camus, is that we can enjoy coming back down the hill. And we can, little by little, we will make this progress over time. This is going to be done systematically, so we can look at these 1.5 million species, but if we look at just the number of families in the Tree of Life, we can do those 9,000 within a couple years. We can get the families, then work our way down to the genus level. This is a 10-year project. We're now only in phase 1 since 2018. EVP has raised a nice amount of funding to get started. And the Moore project is one of those, because we'll be adding 50 just within our own lab, 50 species of invertebrates. We want to raise the quality of these genomes. We want to make these better, and they will continue to increase. So just I'm going to skip this because I know we're running out of time. I know we did the insects. And just lastly, these are just some examples here of some genomes that are going online, like the copepods. These are getting better. Genomes can tell us about maybe which types of species can solve some of our pollution problems, like being able to eat plastic or mealworms or polystyrene, styrofoam. And it turns out this is also helped by a microbiome that lives within its gut. This is a genome of the octopus. There are now 2 octopi that have been sequenced, and they're finding a high activity of neural genes and neural network genes. So we can learn from these, again, simpler animals. And these are, again, just going forward. The last thing here is that GIGA is really focused on training. So we want to expand, as I started off with, expand the capability of computational biology, because it's going to require, the use of programming, scripting, and line commands to handle these large amounts of data sets. So we've sponsored several workshops. We welcome all types of biologists, students to join us. We've now become a nonprofit group this past year. I've been elected as president of the group for another year. And then we want this to be a community, a grassroots community, bottoms-up community that's driven by the membership to create this cooperative, collaborative village to try to understand all of these different species of invertebrates and their genomes, as well as transcriptomes. So it's going to be an ongoing project. We've had some really nice progress here in just the 7 years that we've been around, and we hope to continue this further. We have about 300 members from multiple countries around the world. We want to expand that more into Asia, and membership is free right now. So you can contact me if you're interested. There's the website. There's a membership form there, and it's all part of evolution, biodiversity, and genomes. So that's kind of a whirlwind of some of the work that we're doing in my lab, but I'm open to questions. Close that.
Joe, that was awesome. I mean, it really was a very, very well done presentation, and I love learning more about your lab every time I talk to you. And this was a great opportunity for it. And if people have questions or in the audience, we still have about 22 participants in here. So if anybody has some, wants to contribute or ask some questions specifically, please jump in here. Nicole will moderate it.
Mm-hmm.
Nicole, you got it? Oh.
Any so far?
We haven't received any questions just yet. Oh, I think we just got one. Okay. So this question goes all the way back to the beginning, but is the certificate program something that you need to apply to in addition to the program that they're already in? I'm not sure which program that would be, but.
It's part of the biology program, and you can do that separately, or you can take these courses along with, say, if you were in the master's program, you can take those depending on which master's program you're in. So you can take them as electives, or if you're not in a master's degree program, just take it until you feel like it's useful or you want to expand that further. So you don't have to be in a master's degree program yet. So it's
okay. Thank you. Another one is, where does most of your funding come from? And is it hard to get funding for microbiology?
Oh, that's a good question. Yeah, all funding is pretty competitive, but we've been lucky to get right now some funding for the algae project, which is a big concern in Florida. So we're writing grants as PIs. We're writing grants every year to try to get funding to support students in the lab. Currently, the algae grant is supporting 2 graduate students, and they learn methods like running the Illumina sequencer we have in our lab, doing the data analysis, and then we look at various ways to fund students, such as TA ships. You can be a TA, and you can get a voucher for some tuition that way, as well as get some support for that's semester by semester. And some students also, I've helped them try to get independent funding when they write for grants on their own, which is also a good way to learn how to write a proposal. So that's another thing. So microbiology is actually pretty hot right now. So there's been successful grant opportunities in microbiology, especially now with COVID-19. A colleague of mine just got funding from local agencies to look at COVID or coronavirus in wastewater. So that's a recently funded project.
That's fascinating. Heidi in the chat asked if you can get the certificate if you're in the the MS in marine biology program.
Oh yeah, it's open. So that'll be, some of those could be elective courses. So you just have to kind of work out with our administrator what it might be, those 2 extra courses, because they're not part of the marine science per se. So that'll just be an extra couple courses to add to your master's degree. And we will work with you on that, and what would be considered an elective as well. But it's considered a separate certificate. There's internships or externships available. We haven't. We need to kind of maybe highlight that more. Try to develop these external connections with local biotechnology groups, and that's something we can probably do better in the future. I have several colleagues who have commercial laboratories, and sometimes they are looking for just some help. So you can get you can get some course credit for doing a semester externship. You can learn a technique while helping that commercial lab, and then get some experience in that lab. So that's something that that is available, is an externship.
Awesome. One more question from our Q&A section. Is the genomics offered next semester for the MSMS the same as the genomics required for the certificate?
Yes, yes. Currently, I've been the main professor teaching that graduate genomics course. So we try to have it, it's the credit is going to go towards the certificate if you just went to the certificate or the degree. So yes, I'm teaching it next semester. I'm trying to get it back into sync, and I'll probably offer it again next fall as well. But I went on sabbatical a couple years ago, and then it really should try to come before the bioinformatics class, which Dr. Ozga teaches. Bioinformatics is a more practical course to do the actual exercises. So, but you can still do both or take them together. But I should, if we can work that out, we'll be offering genomics both semesters. But we'll be happy to see you and other students next semester.
Not to put a plug in there, Joe, but Dr. Ozga is speaking next Thursday as a virtual lecture series. So if anybody would like to attend that too as well, we'll send you a notification. So
Great, yeah, he's been great in filling in and picking up that course since he's a new faculty.
Any more questions for the audience?
Not at the moment. If nobody has any more questions right now, but for whatever reason you think of some more later on, we do plan to follow up with everybody that attended today. So, if you have something in mind and you can't quite form it into words, we'll be reaching out to you, and then you can always reach out back to us with whatever your questions are.
Awesome. Well, thanks, Joe. I really appreciate it. It was excellent.
Thanks, Matt. Thanks, Nicole. Yeah, and you just have them. They can contact me if you want to give my email out. I'm definitely happy to talk about our program, research opportunities.
Excellent.
Appreciate it.
We appreciate it, and we'll talk soon, Joe. I'll be sending you an email tomorrow. And Nicole and I were very happy and appreciative that you're willing to speak to the audience.
Yes, no worries.
Thank you.
All right. Have a great night, everybody.
Thanks. Have a great night.
Bye, everyone.